Okay, let's unpack this. Imagine a weapon so subtle, so, sophisticated, that it targets the deepest levels of your biology.
Right.
The actual communication signals that keep your heart beating, your neurons firing, your mood
Elaborate
stable.
background processes you never think about.
Exactly. And when this weapon succeeds, the victim, well, they don't die of a recognized virus or some chemical attack.
Hmm.
A pathologist examines the body, and they conclude the cause was a sudden, natural yet catastrophic internal event, maybe a massive stroke or an aggressive autoimmune collapse, a
Yeah.
ghost weapon.
Please, no forensic trace.
None.
It's the ultimate form of plausible deniability. It's really what intelligence services dream of.
Right.
This capability, it was precisely the strategic objective driving decades of advanced covert biological warfare research.
It seems like a huge shift.
It represents the pinnacle shift in biowarfare, really, moving the battlefield from, say, external infection and rapid, very conspicuous mass death.
Right, like anthrax or something.
It's exactly to silent, targeted, internal biological manipulation.
Our mission today is a deep dive into this, frankly, terrifying corner of molecular warfare. We're focusing on what are called regulatory peptides, RPs for short.
Yeah.
These are tiny biomolecules, small chains of amino acids that are absolutely essential for life. They act as the crucial signaling molecules within our central nervous system, our immune system.
They're like the body's internal text messages.
That's a great way to put it. They dictate function at the most minute level.
And that very power, the role as these potent, essential signaling molecules is, well, that's what makes them profoundly dangerous when manipulated.
How so?
Their biological potency means that a tiny, precise input can generate a massive systemic output.
So a little push causes a big fall.
A very big fall. If you can hijack the body's primary communication network, you can dictate catastrophic outcomes, from paralysis all the way to a complete psychological breakdown.
The sources for this deep dive are just chilling. We're pulling back the curtain on research that comes primarily from the former Soviet Union's extensive and highly specialized, deeply clandestine offensive biological warfare programs.
The BW programs, yeah.
These programs were dedicated specifically to the exploitation and genetic engineering of these neuroregulatory peptides. This wasn't some side project for them.
No, not at all. This was a dedicated, application-focused development program aimed at core political and military objectives.
It's crucial to understand, then, that this level of research, it kind of bypassed the traditional biological threats, didn't it? Things like weaponized bacteria or mass casualty viruses.
Oh, completely. The objective wasn't immediate, widespread death that would signal a biological attack. That's too noisy.
Right.
The objective was surgical stealth, targeted assassination, or mass psychological incapacitation without any hope of attribution.
They wanted weapons that could start and finish a conflict before anyone even knew a weapon had been deployed.
That's it, exactly.
So the big question we're grappling with throughout this deep dive is this. How exactly do scientists turn the body's own natural signaling system into an untraceable, customized weapon of assassination or mass disruption? And maybe more importantly, given the incredible advances in genetic engineering since the fall of the Soviet Union, what does this specialized, technically advanced historical research reveal about the capabilities and the inherent threats of molecular bioweapons today?
The answers, I think, reveal a kind of molecular arms race that is often entirely invisible to us.
Okay, so let's ground ourselves in the basics first. Regulatory peptides, RPs, we probably need to go beyond just calling them signaling molecules.
We do.
To really appreciate the danger, we have to grasp just how fine-tuned their normal function is.
Okay, so think of RPs as the VIP messengers in your body. They're often classified as neuropeptides or peptide hormones.
What's the difference?
They're far more specific and fast-acting than, say, a slow-release steroid hormone. We're talking about absolutely critical processes.
Like what?
They regulate gut motility, they control feelings of pain and euphoria like endorphins, which are a type of peptide. They manage stress responses, like corticotropin-releasing hormone, and they dictate the speed of nerve signal transmission. They are, the molecular rheostats of human existence.
So the moment you introduce an engineered version or you just overwhelm the system with an unnatural quantity of a natural RP.
You introduce instability, chaos, really.
And that is the definition of their dual-use potential.
Exactly. They are fundamentally a double-edged sword, and it's inherent to our own biology.
Right.
In clinical settings, we study RPs for therapy. How can we use a specific peptide to reduce chronic pain or manage autoimmune responses?
Just for medicine.
But because they're so potent and so specific, the line between a therapeutic intervention and a devastating pathological event is extremely thin.
It's a little too much of a good thing.
A lot too much. The mere overproduction of a naturally occurring peptide, say, a powerful inflammatory peptide like substance P, that could lead to an acute, body-wide inflammatory cascade that mimics septic shock.
Wow.
And it would do that without introducing a single foreign microbe. The result is severe pathology, but it's driven by the body's own mechanisms in overdrive.
That distinction that the pathology is caused by the body overreacting to its own molecule, that's what makes the appeal of covertness and lack of attribution so overwhelming for intelligence agencies, isn't it?
Absolutely.
If the attack just looks like a severe, sudden natural failure, the attacker is invisible.
They function as the perfect stealth weapons. For groups like the KGB, this was the holy grail.
Of course.
How do you assassinate a foreign leader without triggering an international incident or revealing a prohibited weapon?
You make it look like he just had a heart attack.
You engineer a death that looks like a massive coronary event or a fatal aneurysm. A pathologist performs an autopsy, finds evidence of widespread vascular collapse or acute neurological damage, and logically, reasonably, attributes it to natural aggressive disease progression.
So the whole mechanism of covertness isn't really about hiding the damage.
No.
It's about ensuring the damage fits a natural disease profile.
Precisely. If the weapon is based on manipulating a molecule the body already uses, all the standard forensic tests designed to detect foreign chemical compounds or engineered infectious agents,
They fail.
They fail immediately. This ability to mimic natural diseases not only aids clandestine operations, but it also, potentially evades the scrutiny of the Biological Weapons Convention.
The BWC.
But if you cannot prove an engineered or prohibited agent was used, declaring a violation becomes functionally impossible.
It's the perfect
Attack. This ability to manipulate the host biology to self-destruct is the hallmark of the most sophisticated covert BW research programs.
So if the goal is this level of deniability and disruption, then the central nervous system, the CNS, that becomes the obvious highest value target.
It has to be.
Targeting a CNS isn't just about killing quickly. It's about guaranteed catastrophic failure, or maybe even worse, psychological control.
Targeting neuroregulatory peptides is the ultimate objective. The peptides they researched were specifically those that could alter mood, trigger devastating psychological changes, or, immediately induce catastrophic physical failure rooted in neurological collapse.
What kind of failures are we talking about?
We are talking about agents that could induce paralysis by overwhelming motor neurons, cause massive hemorrhagic strokes by disrupting vascular control peptides in the brain,
Oh, wow.
or trigger sudden heart attacks through systemic nerve signal corruption.
You bring up mood and psychological changes. This goes far beyond the typical concept of a lethal weapon.
It does.
It suggests a capacity for targeted psychological warfare, making a political figure paranoid or unstable or just incapable of coherent thought during a critical negotiation.
Absolutely. The research focus on neurotropic agents was tied directly to creating agents that could induce acute, dramatic psychological episodes.
For example?
Say, overwhelming aggression, profound apathy, or suicidal depression.
So a politician who suffers a sudden psychological collapse and just retreats from public life or commits suicide.
Achieves the military or political objective without the slightest hint of external intervention.
The weapon works by maximizing the disruption of essential human cognitive functions.
Decision-making functions. It's an information attack at the level of the brain's internal circuitry.
And what really stands out to me here is the level of biological literacy required. This isn't just, dumping a poison into the water supply.
Not even close.
This is molecular architecture for destruction. It requires immense specialization and understanding of the most sensitive parts of human physiology.
It is a massive intellectual leap. When we talk about RPs causing strokes or paralysis, we're talking about the targeted disruption of incredibly complex biological feedback loops that operate minute by minute. This knowledge base is light years more specialized than traditional pathogen weaponization. It requires synthesizing the ability of the body to regulate itself and then turning that regulatory ability into a self-destructive command.
Okay, let's take that theoretical architecture and apply it to the operational reality of the Soviet Union, because this is where it gets really interesting. We're moving from the molecular theory to the specific, named, and highly prioritized programs that were operating under their third directorate.
And the fact that they established separate, compartmentalized programs with distinct code names That underscores the strategic priority they placed on developing these stealth capabilities.
This wasn't just some academic curiosity.
No, this was core state security doctrine.
So let's start with program Flute. The oversight for this, as the sources detail, it wasn't the Ministry of Defense.
Which you might expect.
Right, which might handle mass casualty agents. It was the Ministry of Health's third directorate.
And that institutional camouflage is a crucial piece of the puzzle.
How so?
Embedding offensive BW development within public health or civilian research infrastructure is a classic move. It's designed specifically to evade internal bureaucratic oversight and, of course, external international scrutiny.
And Flute's stated goal?
Developing psychotropic and neurotropic germ agents.
And the application reveals the real target. These agents were intended specifically for the KGB's special operations.
The political assassinations, the destabilization campaigns.
What's commonly referred to as wet work.
Yes, and this connects directly back to that psychological dimension we just discussed. The goal of Flute was to create agents that would induce acute, targeted alterations in mood and behavior.
So the desired effect, it wasn't necessarily immediate death.
Not always, no. It was temporary or permanent psychological incapacitation. We are talking about the ability to induce, on command, extreme paranoia that leads a target to make catastrophic public errors or chemically induced decision paralysis at a moment of crisis.
The ability to inject a temporary high-dose peptide that just floods the system, forcing a psychotic episode or a period of severe depression that leads to a subsequent accident.
It's horrifyingly effective for covert political operations.
It is.
And the neurotropic mechanism means these agents are designed to efficiently cross the blood-brain barrier.
Which is the brain's defense shield, right?
Exactly. A highly selective membrane designed to protect the CNS. RPs are ideal candidates for this because many natural RPs are themselves neurotransmitters or neuromodulators, or they have carrier systems that naturally facilitate CNS access.
So they have a built-in key.
A key, a delivery truck, yes. By flooding the system with a massive targeted peptide, you introduce a chemical chaos that disrupts the extremely sensitive balance of neurotransmission. The entire premise of Program Flute was harnessing this access for strategic control.
Okay, now let's move to Program Bonfire. This one focused less on psychotropic disruption and more on overtly toxic physical destruction.
Still using the stealth peptide concept, though.
Right.
Bonfire was the program tasked with finding peptides that could cause maximum untraceable physical damage. And what's fascinating here is that the scientists working on this program, they successfully identified and chemically named a specific regulatory peptide derived from this work.
And what was it called?
The myelin toxin.
The name itself is immediately revealing. We absolutely need to spend some time on the mechanism of this myelin toxin because it sounds like it targets the very infrastructure of the nervous system.
It does. Let's use that analogy you mentioned earlier. Think of your thousands of nerve fibers, the axons, as high-speed electrical wires.
Okay.
These wires transmit every signal, from conscious thought to muscle contraction. For these signals to travel fast and efficiently, they require insulation.
And that insulation is the myelin sheath.
Right, a fatty protective layer wrapped around the nerve fibers.
So the myelin toxin is designed to compromise that insulation.
To strip it away. When the myelin toxin is present in massive, unnatural quantities, it destroys that protective layer in a process called demyelination.
And that's catastrophic for the nervous system.
It is. When a myelin is damaged, the electrical signals slow down, they scatter, they short-circuit, or they stop entirely.
Like frayed wiring.
Exactly like frayed wiring.
So what are the clinical outcomes of that kind of rapid systemic demyelination?
Oh, they are devastating. And they mimic some of the most feared natural neurological diseases. We are talking about acute, rapid onset of severe neurological dysfunction.
So it's just
Profound muscle weakness, loss of coordination, debilitating sensory issues, and ultimately paralysis.
And critically, these symptoms would clinically resemble what?
An aggressive, acute flare-up of multiple sclerosis, MS, or perhaps Guillain-Barré syndrome, a scenario where the body is already known to attack its own myelin.
Wait, hold on. So if the victim were to die from, say, respiratory failure linked to paralysis induced by this toxin, would the pathologist look at the nerve tissue, see the demyelination, and just conclude?
It was an aggressive, natural autoimmune condition. That is the precise objective. The pathologist would observe widespread nervous system damage consistent with known, sudden neurodegenerative or autoimmune conditions. They would find evidence of the demyelination process, but
no weapon.
Without finding an easily identifiable foreign bacterial toxin or chemical residue, they would attribute the death to an unfortunate natural cause. The damage itself, being an aggressive mimic of a natural disease, completely conceals the weapon.
The discovery and synthesis of a specific molecule like myelin toxin clearly demonstrates that the Soviet BW program was not just focused on bulk pathogen production.
No.
They were deeply invested in the cutting edge of molecular biology and neuroscience to create these highly tailored, untraceable agents.
They moved from carpet bombing to surgical strikes at the molecular level.
And once they had isolated these potent peptides, from the neurotropic disruptors of Flute to the demyelinating poisons of Bonfire, the next inevitable and terrifying step was figuring out how to mass produce and deliver them.
Efficiently, yeah.
And this is where the intersection with genetic engineering became unavoidable. The problem with RPs for military application is, I guess, a purely logistical one, right?
It is.
There are complex molecules involved in these intricate biological feedback loops. They must be difficult and expensive to synthesize in the massive quantities needed for even covert operations.
That's a significant technical hurdle. That logistical constraint is what initially protected us from widespread peptide weapons.
How so?
You can discover the deadliest peptide known to science, but if you need a year and $1 billion to produce enough for one dose, it's not a viable weapon.
Right, it's not practical.
So the solution, which they arrived at through the rapid advancement of molecular biology in the 80s and 90s, was to outsource the production.
Outsource it to what?
To microscopic, self-replicating organisms, bacteria, and viruses. They turn them into instant peptide factories.
So how did they use genetic engineering to overcome the production challenge for, say, the myelin toxin? Can you describe the molecular steps?
Sure. The process is meticulous, and it begins with the blueprint. Step 1 was synthesizing the specific gene, the precise sequence of DNA that contains the instructions, or the code, for producing the toxic myelin peptide.
They had to chemically write the blueprint for the weapon?
They did. This required complex oligonucleotide synthesis and often techniques like polymerase chain reaction, PCR, to amplify and verify the correct sequence.
They write the code. Okay, what's step 2?
They artificially reproduce and stabilize this toxic gene sequence in the lab, often splicing it into a small, circular piece of DNA called a plasmid.
A plasmid?
It acts as a shuttle. It ensures a stable, abundant supply of the blueprint. This step is about making the blueprint durable and transportable.
Got it. And then the delivery system, step 3, that's where the gene is inserted into the infectious vector.
Precisely. They inserted the synthesized gene into an infectious agent, typically a well-understood, robust bacterium or virus, the vector.
The delivery truck.
These vectors are biological delivery trucks, yes. Chosen specifically because they are highly efficient at invading human cells and hijacking the cell's internal machinery.
Turning the host cell into a forced peptide-producing factory.
Exactly. Once inside the victim, the vector forces the host cells to start mass-producing this new foreign product, the myelin toxin. The victim's own cells essentially self-destruct by manufacturing their own poison.
That is a terrifying escalation, moving from just synthesizing a chemical compound to creating a genetically altered bioweapon that replicates and delivers the toxin systemically within the victim.
It's a massive leap in capability and, well, proliferation concern. The sources explicitly note the short, logical step considered by researchers once this gene insertion technique was mastered.
Which was?
They considered moving the myelin toxin gene insertion from relatively low lethality bacteria like Yersinia pseudotuberculosis.
Which causes what exactly?
A relatively common, treatable gut infection.
Okay.
To inserting it into Yersinia pestis.
Yersinia pestis. That's the bacteria that causes the plague.
Yes, the plague. This fusion of capabilities is extremely alarming.
Because you're creating a hybrid weapon.
You are. A weapon that possesses the historical lethality and aerosol dispersion potential associated with plague, a traditional BW agent, and is simultaneously a devastating, rapidly produced neuroagent.
Wait, so the victim is not only infected with the plague, but their body is also immediately and systemically producing a neurotoxin. What would that do to the prognosis?
It would dramatically reduce the window for intervention.
I'd imagine.
Even if standard medical countermeasures, antibiotics, were rapidly effective against the bacterial infection itself, the damage caused by the massive rapid production of the myelin toxin inside the host would be immediate and severe.
So even if you cure the plague, the neurological damage is already done.
It would likely cause permanent, irreversible neurological damage, severe incapacitation, or death before the infectious process could even be stopped. This truly exemplifies the power of advanced molecular biology to create synthetic pathogens with tailored, multi-layered effects.
But the research didn't stop at just synthesizing and inserting new toxins.
No, it didn't.
Advanced programs, likely realizing the inherent difficulty in controlling the replication of a hybrid plague agent, focused on an even more insidious, truly covert strategy, triggering autoimmunity.
This is where the concept of the ghost weapon reaches its absolute zenith.
How so?
Instead of poisoning the system with an external toxin, the goal shifted to reprogramming the body's own defense system to attack the host. It's biological self-sabotage, perfectly replicating natural disease.
How do you achieve that level of deception at the molecular level?
This requires exploiting the body's self-recognition system. Researchers engineered viruses to carry and express genes that code for protein fragments of myelin.
The same material from the myelin sheath?
The very same. The virus infects the body, and instead of producing a toxin, it forces the cell to express these myelin fragments as if they were viral components.
So the body's immune system, which is designed to identify anything foreign,
sees these fragments and begins to mount a massive targeted response.
So the immune system is essentially trained or programmed to believe that myelin, its own crucial nerve insulation, is the enemy.
That is the sophisticated goal. The process is called molecular mimicry. The body launches a full-scale, devastating autoimmune response against its own, naturally occurring myelin or other critical tissues.
And the outcome is a self-inflicted, devastating pathology that clinically mimics what?
Devastating chronic autoimmune diseases like lupus, rheumatoid arthritis, or aggressive MS, but induced acutely and intentionally by the engineered vector.
An induced chronic illness that pathologists would never trace back to a weapon.
Never.
Because the mechanism is fundamentally the body attacking itself. The presence of the initiating viral vector might be gone or clinically irrelevant by the time the pathology manifests fully.
Exactly. The attack is entirely plausible as a natural event, a tragic, aggressive autoimmune disorder. There was no foreign chemical residue. There is no easily recognizable foreign bioweapon footprint.
There is only the immune system, the body's security force turning violently against its host.
All due to an engineered molecular deception. If we connect this back to the initial search for non-attribution, the ability to manipulate the fundamental mechanism of self-recognition is arguably the most terrifying and hardest to track bioweapon capability.
Understanding the molecular science is only half the picture, though.
Right.
The other half is understanding the institutions that funded, executed, and frankly protected this research. These were not fringe labs.
No, these were high-level, state-funded centers.
And the sources identify some key players involved in developing these psychotropic agents and peptides.
Yes, we have to focus on two institutions in particular, the Moscow Institute of Immunology, which provided foundational research, and the Research Center of Molecular Diagnostics and Therapy.
And it's the second institute that was explicitly identified as being intimately involved in program Flute.
The psychotropic and assassination research, yes.
The name Molecular Diagnostics and Therapy is the perfect camouflage, isn't it?
It is.
It sounds like a cutting-edge medical facility, which allowed offensive work aimed at KGB special operations to hide in plain sight under the guise of legitimate medical research.
And this leads directly into the critical issue of oversight and dual-use funding in the post-Cold War era. The source material highlights a really staggering ethical and security vulnerability. The Research Center of Molecular Diagnostics and Therapy, a facility deeply implicated in clandestine BW research, specifically developing mood-altering peptides for covert operations, was later involved in U.S.-funded grant programs.
Wait, say that again? International funds intended for the peaceful transition of science were potentially flowing into the same physical infrastructure.
And possibly employing the same personnel.
Who had just been developing tools for political assassination. That sounds like a catastrophic failure of non-proliferation mechanisms.
It raises profound questions about the efficacy of international oversight. How do bodies charged with non-proliferation properly vet and oversee facilities that have a demonstrated recent history of highly specialized offensive military work?
I mean, the expertise doesn't just disappear.
Exactly. The expertise required for this type of molecular warfare, the genetic engineering skills, the knowledge of regulatory peptide systems, the specialized equipment, it does not simply vanish when a program is officially shuttered.
It transitions.
It transitions, often seamlessly, into civilian research, presenting a persistent leakage risk. The dual-use nature of the research means the same scientific inquiry used to find a new drug can be immediately reapplied to create a highly specific, deadly covert agent.
And the research conducted by these institutions wasn't just scientifically advanced. It was allegedly carried out under conditions of immense ethical depravity. The sources detail serious, specific allegations made by Russian scientists regarding the ethical breaches at the Research Center of Molecular Diagnostics and Therapy.
This is arguably the most morally corrosive revelation in the sources.
What's the allegation?
The allegation is that the Institute crossed a profound moral taboo, testing its peptides and mood-altering agents, specifically those developed for the Flute program, designed for clandestine political control on patients confined to an adjacent hospital.
They were testing agents designed to induce paranoia, severe depression, or decision paralysis on unknowing captive human subjects.
Yes.
What was the institutional goal behind this human experimentation?
The goal was clinical validation, which is essential for any operational weapon. It wasn't enough to know the peptide worked on a primate.
They needed to know the precise, real-world effect on humans.
On human mood, behavior, and cognitive processes. Program Flute required agents that achieve psychological incapacitation or control, and that requires empirical clinical testing on humans to understand dosing, latency, and manifestation of psychological symptoms.
And the patients in the adjacent hospital.
Likely vulnerable and non-consenting, provided the necessary testing pool to confirm the operational utility of these psychotropic agents.
That level of institutional control, where a major research center collaborates with a hospital to utilize its patients for offensive weapon validation, it speaks volumes about the complete lack of ethical constraints surrounding the Soviet Third Directorate.
It confirms the extreme lengths to which these programs went. When you look at the sophisticated science, the genetic engineering, the myelin toxin discovery, it's clear they had immense technical capability.
Right.
But coupling that technical mastery with this profound willingness to violate human rights on this scale demonstrates the immense danger inherent in specialized state-run bioweapon programs. The scientific results are inseparable from the ethical framework, or lack thereof, within which they were produced.
This history of profound ethical breaches and institutional entanglement makes the broader proliferation concern exceptionally urgent today, especially as technology miniaturizes.
The overarching trend we see today is the increasing sophistication and understanding of the human immune defense system and infectious disease mechanisms, which is fueled by the rapid global accessibility of genetically engineered vectors.
So we're in an age where the molecular knowledge that was once restricted to hyper-secret Soviet institutes is now
available in labs worldwide.
And this brings us back to the stark reality of the dual-use dilemma. The very tools that allow us to develop breakthrough gene therapies, or target specific peptides for cancer treatment, or develop new classes of vaccines,
are the exact same tools that allow a rogue actor or nation-state to create more virulent, untraceable bioweapons.
Molecular biology is inherently neutral, but its application is not.
That's right. If a young scientist learns how to synthesize a gene and insert it into a virus to cure a genetic illness, they simultaneously possess the knowledge to synthesize the myelin toxin gene and insert it into a bacteria for an assassination attempt.
And because this research deals with manipulating molecules in code, rather than stockpiling large quantities of visible pathogens, it becomes extraordinarily difficult to detect and control.
Especially through traditional arms control verification regimes. The weapon is not the facility. The weapon is the intellectual property, the genetic code and the protocols, and the readily available specialized equipment.
It just underscores why the institutional context, specifically the failure of oversight when facilities transition from offensive military work to ostensibly civilian research, remains an acute security vulnerability.
The leakage of personnel and specialized knowledge is the biggest non-proliferation challenge we face regarding molecular bioweapons. We're dealing with capabilities that are becoming smaller, more potent, and exponentially harder to monitor as molecular technology advances.
And the history of programs Flute and Bonfire serves as a critical warning.
It does. They show that the ultimate goal of offensive BW research is not mass destruction, which is easily attributable, but perfect stealth and tailor-made, non-attributable disruption, leveraging the most sophisticated understanding of human biology possible.
So what does this all mean? We have undertaken a deep dive into an extraordinarily specialized and deeply disturbing corner of biological warfare history. What the sources reveal is the unique and persistent threat posed by the weaponization of regulatory peptides. Untraceable agents designed to cause catastrophic neurological dysfunction, psychological chaos, or to trick the body into an act of self-targeting autoimmune destruction.
And this capability was historically solved by leveraging sophisticated genetic engineering to overcome the natural production and delivery hurdles.
But today,
Today, those engineering techniques are far more advanced and far more accessible globally, meaning the theoretical threat demonstrated by these Soviet programs has only amplified.
the ultimate takeaway for me is that this research demonstrates a fundamental and terrifying shift in bioweapon strategy.
I agree.
We have definitively moved away from agents that kill quickly and conspicuously, the classic obvious weapons, toward highly complex agents that are designed specifically to conceal the hand of the attacker.
Guaranteeing non-attribution and plausible deniability.
Right.
The threat is no longer necessarily visible mass death. It is invisible, targeted chaos driven by internal miscommunication.
If a stroke, paralysis, or a sudden aggressive autoimmune disease can be weaponized and induced by an engineered peptide or a self-targeting virus, then the entire concept of identifying and defending against a biological attack has fundamentally shifted.
It has. We are moving from defense against external invasion to defense against internal betrayal.
This research forces us to confront the fact that the most potent and likely future bioweapons may not be easily recognizable pathogens,
Right.
but rather the weaponization of the body's own communication systems turned against itself.
And that leads us to a provocative closing thought for you to consider. If the most advanced historical bioweapon research focused on weaponizing the body's own communication signals, these crucial regulatory peptides, what current common pharmaceutical or diagnostic research happening today, particularly in advanced gene therapy or neuroscience, might unknowingly hold the potential for a covert biological application tomorrow?
And if the intellectual property for a perfectly untraceable weapon is just a sequence of genetic code, how can traditional arms control ever hope to govern the future of molecular biology?
That is something substantial for you to think about.